We are very pleased to have Dr. Yaning Wang, who is the director of the Division of Pharmacometrics at in Cedar at US FDA, to speak to us today about regulatory impact of phase two model-informed drug development. We do have the chat window, the Q&A window is where you should submit questions, and we'll try to have a few minutes at the end for him to address some of those. And then do please keep mics muted during the session. I'm going to turn it over to you now, and thank you very much, Yaning.
So first of all, thank you very much for the introduction. Hello, everyone. Today, I will talk about the impact of model-informed drug development during phase 2 of new drug development from a regulatory perspective. As you all know, MIDD is an ongoing new pilot program, or you can consider that as overall drug development strategy. But in order to talk about the impact of MIDD, it's impossible not to talk about an earlier pilot program that's called end of phase 2A meeting pilot program that started right after I joined FDA in 2003. That's almost 18 years ago. Time flies. So in fact, this guidance is still on the FDA website, even though you can consider it's outdated or replaced by the current MIDD pilot program. But when we started this, back in 2004 or 6, a new guidance was generated out of that pilot program, and that guidance is called End of 2A meeting guidance. As you can see from the screen, the purpose of this meeting was to facilitate interaction between FDA and sponsor who seek guidance related to clinical trial designs, especially using the clinical trial simulation and quantitative modeling of prior knowledge, specifically drug, disease, and trial models to help or optimize their later phase trial designs, such as phase 2B and phase 3 trials. And there's a reason why back then we picked end of phase 2A, because that's when the sponsor collected early phase 1, PK, some biomarker data, and also in patients, in the phase 2A phase, some clinical endpoint or some relevant biomarker in the target population. We felt that is the most relevant period. We'll have enough data to have a meaningful conversation, between FDA and the sponsors. So that's when, we started this very specific end of 2A, even though we already had an end of phase 2 meeting, but we felt that that was too late typically for FDA to provide any meaningful feedback for the sponsor to make significant changes if we disagree with their plan. Because typically when the time is already end of phase 2, the sponsor pretty much already made up their mind about how to do their next phase 3 trial design. We felt that was too late. Therefore, we moved this critical meeting into like end of phase 2A phase. And like I said, it was a pilot program. We collected about a dozen examples during this period of pilot and published our experience. As you can see, this was all the cases we did during the 2004 and 2006 cases. These were all from the end of 2A pilot program. And in this paper, we summarized 11 cases, covering different therapeutic areas. Again, these are all end of phase 2A cases. Therefore, as you can see, the key question is all about either phase 2B design or the phase 3 or registration trial design and dose selection. And it covered 11 different therapeutic areas. And back then, since it was a pilot program, we were trying to collect feedback from both the within FDA, because this was initiated from the Office of Clinical Pharmacology and specifically targeting the modeling domain. So we were getting feedback from within FDA, our non-pharmacometrician colleagues, stats or medical colleagues, and also from the sponsors side. As you can see on the left side, this is an average score from both within FDA and from the sponsors, for the 5 different therapeutic areas. And here, 0 meaning, no value, or 5 meaning pivotal. As you can see, the average feedback, varies, even within FDA or between FDA and the sponsor. Overall, the sponsors felt it was pivotal or more towards pivotal. But as you can see within FDA, some of therapeutic areas, they did not feel it was a pivotal. And on the right side, we have another one that is a specific asking the sponsor question whether this interaction, the end of two interaction, changed the drug development plan, from the sponsor side. One, of course, is changed, zero, not. So again, this is average. As you can see, we explained, if it's a one, that means all the responders said, this interaction changed their plan. And if it's like this one is close to 60 some percent, that means the other 40% felt they did not change their drug development plan. And of course, we also include in this paper, very specific feedback comments from both FDA and the sponsor side. As you can see back then, this was, I'm going to show you one example to highlight. It's a very workload intensive kind of review within a very short period of time. Therefore, as you can see, the first comment from FDA side was the reviewers don't have the time to do the sponsors work for them. Doesn't make sense to work this way in light of the vast difference in resources between us, the FDA, and the industry. This was actually a very common questions, especially raised within FDA, because before this pilot program, this is one type of IND review. Therefore, it's not common for people to spend so much time on an IND review. Therefore, this was a very common feedback we got, from the FDA. And in fact, this played a very important role in the next, MIDD pilot program that was supported by the PDUFA 6. We needed more resources to hire more people, to have more people to dedicate their time on this type of IND interactions. And also, this one, the second one, is more positive. Most people consider it's very helpful. I thought it, and more than that, is because the previous drug experience, I strongly believe we were able to apply our lessons learned from the previous drug to this new drug development strategy. Even though this one you can believe it's applied to any IND meetings, all the FDA reviewers or the decision makers, they tend to apply what they learned from other therapeutic areas or other drugs from different sponsors to this new one. But this one was particularly relevant. And this one actually is probably from one of the FDA reviewers, as the sponsor's analysis was sufficient to conclude the dose selection was appropriate. Modeling by FDA had minimal impact. And this, of course, is one of the examples. We have so many examples. This reviewer worked on one example. The sponsor did excellent work. Therefore, the FDA additional work did not, significantly change the conclusion. And I want to read through all of them. You can see also from the sponsor side. And the last one, again, the pharmacometric assessment must be very resource intensive, but it appears to be one of the most useful tools that FDA has to enhance drug development. This again highlights the limited resource we had back then to deal with this kind of a very resource intensive review. And for the sponsor side, I think most of them are positive, because, and there's a reason why this dozen of sponsors chose to test this pilot, because they saw a need and they clearly got what they wanted, through this interaction. And most of the comments are very positive. And in addition to our publication to summarize our experience, through that dozens of cases, the external experts, for example, Dr. Raymond Ousley and Dr. Don Stansky and John Orloff, they both, these two papers from the outside also highlighted the values. One is saying they are from the government, they really are here to help you, and the communication with FDA, the third of the new model for drug development. Again, they highlight this, but again, from the FDA internal experience, we felt a stress given our limited resource. But overall, it was positive, especially from the outside feedback. I'm going to use one example to demonstrate why it was resource intensive. This is an old example, like I said, it's almost 18 years since we started this pilot program, but it's still one of the very few examples, or best examples, I would say, that implemented what we call a disease model, a drug model, and trial model. All three components were included in a very nice way to help design phase 2B and the phase 3 trial. by analyzing all the existing data up to end of phase 2A. So the model involved, it's an antiviral drug. The disease model was a viral dynamic model. For the drug model, actually, it's complicated because this drug was supposed to be added on top of Calitra that was already approved a drug for HIV. So among the three components, the Calitra has two components, and there are PK interactions because lopinavir and ritonavir, one of them is the PK booster. Ritonavir is the PK booster. The other one is the antiviral drug. And it turned out the ritonavir is going to also increase the drug exposure for this drug, the new drug. So therefore, there's PK interaction between lopinavir and ritonavir in the approved drug, and there's also PK interaction between ritonavir and drug X. And at the same time, since the drugs are working on different targets, I will show that on the next slide, there's also pharmacodynamic interaction. So all these are nicely incorporated in this drug model. And the last component is the trial model. HIV trial compliance, dropout, there are major issues in terms of affecting the final success rate. So the viral dynamic model back then, it was already used by another sponsor. So we learned, like I said, learned from a different sponsor. We applied this model to a new sponsor. And of course, these days, this type of model is commonly used not only in HIV, but also in other virus model as well. So simply speaking, the CD4 cell will be affected by virus to create two groups, active infect T cell and latent infect T cells. And the latent cells can also be reactivated to the active infected cells. And then the active infected cell can produce more virus. And this process is inhibited by the approved drug, Calitra. That's how the drug works, working on this step. And this new drug, drug X, is working on a different step. That's blocking the virus, affecting the CD4 cells. So as you can see, this is how the PD interaction was incorporated into this mechanistic disease model. And of course, there are PK models for all the three components. One is, and of course, among the three, like I said, ritonavir's exposure will inhibit enzyme that's responsible for the metabolism of lopinavir and the drug X. So that's the PK interaction. Then on top of that, there is this compliance model, which was developed based on analyzing historical HIV trials. The top one showed one trial result that showed the relationship between the proportion of patients or proportion of patients taking, what percentage of the prescribed drugs. Some patient could take up to almost 100%, but some patient only take 40%. This is a compliance distribution. And at the same time, show the mean and the standard deviation. And the bottom plot shows the correlation between the mean patient adherence and the standard deviation to show, given the different sample size and different the means compliance, the standard deviation, the variability among the individual is also different. So this simple statistical model, there's no mechanistic reason to explain why patients are not taking this. This model was not linked to, let's say, toxicity or lack of efficacy for patients not following compliance. It's a purely statistical model to describe the random behavior among the HIV patients within the trial. And the last component is dropout model. The previous speaker already mentioned. So here we also applied a dropout model by analyzing some large phase 3 trial from another compound. That's the advantage of the FDA database. We already have data from a phase 3 trial to observe what would be the dropout pattern in a phase 3 trial. As you can see, there were two major phases, the first 4 weeks then after that up to 48 weeks. 48 weeks is the end of the trial. After that, that's the follow-up. So we just took a very simple, like a stepwise linear model. The first part is one linear model, then between 4 weeks and 48 is another linear model to describe this simple dropout model pattern. And we actually implemented two different scenarios. One is, depending on the dose, this one actually was linked to the toxicity and also the dosing regimen. And the first scenario was, we have 30% dropout at the week 48, then you take half, that will be the dropout at week 4. And then we have a higher dropout at 20 milligram BID because of the toxicity. There was some GI toxicity. And for 40 milligram QD, we believe, the sponsor proposed that there may be a lower dropout rate because this is a QD regimen. The other one, tools were BID regimen. But given that we did observe some QD related toxicity in the early phase one, so we also assessed another different scenario that is higher dropout, for the QD regimen. So all these scenarios, together with the earlier PK model for three components connected through the interaction. Then we have a virodynamic model through the PD model. And then, of course, everything also was connected because of the different targets of the two drugs. And then we add adherence to model the human behavior or the patient behavior in terms of taking the drug. And the last component is the dropout, in terms of how many people drop out. Again, this one, qualitatively, we incorporate the exposure through the toxicity, but it's not a exposure toxicity driven dropout model. So after this exercise, again, within that very short period of time, we were able to build the model again by learning from another sponsor and apply to this specific case, and conducted enough clinical trial simulations to make the following recommendation. And finally, we felt that the more frequent regimen is a better regimen than the lower BID regimen is more likely to have better efficacy instead of the higher dose QD. And the sponsor was trying to make a decision right after they started this trial, 4 weeks. And we felt that it was too short to select the dose, especially if we want to differentiate the efficacy among different dosing regimens, again, on top of another already very potent drug. And we asked the sponsor to wait a little bit longer until week 20, 12 and 16 weeks to assess the efficacy in order to pick the right dose to start their phase three, and continue this until week 24 to confirm they pick the right dose. And we felt that the margin for further improvement of efficacy is not that large, to differentiate different dosing regimens, because the underlying background regimen is already pretty strong. And we felt the ultimate factor that will determine which dose will be the best may be most likely to be driven by the toxicity. So these recommendations were delivered to the companies, and they were able to take our advice into consideration for their next phase. So that's basically a history where we started this. That pilot agreement, like I said, triggered discussion between FDA and pharma to provide more resource, more money through the PDUFA program, so that FDA can hire more experts in this domain to restart this program. Yeah, we had a guidance, but after that, because of the resource, it slowly, I guess, became outdated until, the PDUFA 6 came out. In fact, in this 5-year kind of period, pharma provided additional resource, especially money, to so that FDA can hire more people. And in return, FDA agreed that for the first bullet point is like we will hire more people to do enhance the science in this area, and also provide the second bullet point, that is we will organize multiple workshops in the field of MIDD to educate the audience or the whole field in terms of the progress in for example, PBPK, exposure response, disease model, and also in the biologic field. The third bullet point is the focus of my second half of the talk. That is, starting from fiscal year 2018, FDA reinitiated this new pilot program. Let's just call it MIDD pilot program. Under this pilot program, we will restarted this IND review, and the numbers of submissions per quarter is limited to 2 to 4 proposals or submissions, because this is shared between CDER and CBER. CBER is our biologic center for vaccine and blood products. And therefore, for each center is 1 to 2. That's the quota after the negotiation between FDA and pharma, they came up with this number. And again, the focus is still dose, duration, and patient selection in a way that can inform regular decision making. But this time we did not limit it to only end of 2A. Any phase of the drug development can come in with pharmacometric oriented or focused discussion. And the last one is, again, in return, FDA agreed, in this 5-year period covered by PDUFA 6, we will publish new guidance or updated existing guidance to further promote this MIDD drug development. So this is now you can see, after all this negotiation, supported by additional resource. And we did hire more people. There are dedicated project managers, and our division also grew from when I joined FDA, the group was a very small team with like 5 people. But now these days, I mean, especially as of today, we are actually the largest division within the Office of Clinical Pharmacology. It's the FTE number is 36. So as you can see, the growth of the people, driven by the workload, and of course, driven by this PDUFA 6, we got additional resource. So this is the new pilot program kind of process. We accept submissions quarterly. So we have a MIDD committee to vote, if there are more than 2 per center or more than 4 for the whole FDA, we have to select, if it's beyond our workload. And some submissions may be rejected for workload reason, and some may be rejected because maybe there were not enough data to have a meaningful discussion. So after the committee vote, for those who are accepted, the sponsor will submit their package. We have 120 days to review the whole package, and also offer 2 meetings to the sponsor to meet with FDA to discuss their questions. After the first meeting, the sponsor can take FDA's feedback and further modify or improve their model or change their protocol or plan, and even come back for the follow-up meeting. And after that, the sponsor can move ahead with their planned drug development plan. So that's the overall process. And 1 year after we started this pilot program, we published a paper to summarize the first year kind of workload, disease area, distribution of the study phases. Here, as you can see, during the 1 year period, we received 15 submissions, and we accepted 13 and rejected 2. Out of these 2, 1 was rejected because the sponsor was trying to do a single arm trial, but FDA was not ready to accept single arm without control. That was the main reason for rejection. And the other one was mainly because of the workload. That's when it happened that 5 submissions were submitted. In fact, these were all in CEDAR. Okay, we were supposed to only handle 2 cases, but we took 4 and rejected the last one. And also, as you can see, the phases of drug development covered from phase 1 all the way to post-marketing. So the entire drug development period was covered, and therapeutic areas covered 7. And in terms of topics like dose selection or clinical trial simulation, all priority topics were covered. And the methodology used also covered everything. And I will have another slide to cover all this detailed pop PK or exposure response or more mechanistic models. And like I said,
just a quick time check. We have just 5 minutes left.
Oh, okay, sure. I'll go faster. So that's the first year. Then this is, of course, up to now, we have 37 total submissions, and we rejected 5, and 4 of them was due to workload issue. And these are the therapeutic areas covered. Now, instead of 7, we covered 13 therapeutic areas, almost covering the entire CEDAR. The most cases came from the oncology domain, but all other areas also have submissions. And then in terms of clinical phase, as I said, it's from preclinical, first in human, all the way to post-approval. And I just highlighted how many cases are specific for phase 2, because today our session is about phase 2. So out of 37, we have 17 that's relevant for phase 2. And sometimes they cover phase 2 and phase 3 together. And then in terms of the goals or the applications, as you can see, most of these are related to dose selection. All of this involve dose selection either for phase 2B or phase 3, and some of them involve clinical trial simulation and the predictive, mechanistic safety. And in terms of methods, like I said, exposure response is the most commonly used methodology because this is about dose optimization. And some of them also use the dose drug disease trial model or only pop PK or even meta-analysis. And some of them involve the QSP, more mechanistic type of models. And again, I summarize these three components only for the phase 2 cases. As you can see, again, even for phase 2, we covered pretty much every therapeutic area. In terms of application, there are mainly dose selection. Of course, that's the main purpose of phase 2. Then in terms of methodology, it covers every possible methodology. In terms of impact, like the previous one, we try to get sponsors' feedback. This one we summarize based on our final recommendation to sponsor. For most of the time, we agree or align on MIDD approach or strategy, and sometimes we alleviate the need for additional studies, meaning the sponsor can waive some studies, and in some cases, they can conduct a smaller study. Like the previous speaker said, it's a more powerful approach so that you can achieve the same goal with smaller studies. So in summary, MIDD or model-informed drug development has a long history of regulatory support, and we have applied models with different complexity to address different decisions at different stage of the drug development. And under the PDUFA 6, we have certainly additional resource and additional momentum to further apply this kind of quantitative methods in more areas of drug development. And finally, I would like to thank, for my former or current colleagues within the Division of Pharmacometrics. This is certainly a pharmacometric-oriented pilot program, but these days, the whole Office of Clinical Pharmacology staff are involved. That's why many people are involved to have many meetings. And we even have dedicated project manager staff supporting MIDD activity, organizing workshops, organizing all the meetings. And of course, this is not only for clinical pharmacology. We have our medical colleagues, statistical colleagues, and sometimes even preclinical colleagues. And then we have dedicated people. And finally, I also want to thank all the sponsors who volunteered or participate actively in this program. Without their participation, either the end of 2A or this one would have not been so successful. Thank you very much.
Thank you. I think that was a very interesting presentation, and it's really nice to see the growth of model-informed development support at FDA. I wasn't at all aware of the size of your group now. That's fantastic. We have seen, even with our own clients, their interaction in model-informed development and through Type C meetings and such things with FDA, and the benefit that can have for them in dose selection. So just a couple of minutes left. Could you give any guidance on what a company can do as they approach the FDA to sort of optimize the chance that their MIDD application would be accepted in? Another nice thing to have heard from you is what large percentage have been accepted in, and really the limit was workflow, but is there, or resource availability. Is there any guidance you would have on sort of what the application should look like to have a company have their MIDD application accepted for review?
I think that's an excellent question. We have a dedicated website. I should have probably put on the slide. On the website, we have outlined the focus areas for this type of meeting to be accepted. Like I said, it's quantitative methods oriented to support key drug development decisions like dose selection, patient selection, or the trial optimization. And one advice I have for those, like I said, our workload allows, we even accepted 4, even though, by the way, these are all CEDAR submissions. So far, all the cases apply to CEDAR only. So we were in theory only can handle 2. But when the workload allows, we even accepted 5, as you can see in the 2020 Q3. And but most of the time, because of the workload or resource, I would say, have the data ready, because I remember one of the cases was too early. They haven't have any like a patient data. It's too early. It's mainly a plan. So for this kind of cases, it's better to have some concrete data, and also you already analyze data to have some concrete recommendation for the next phase in terms of dose selection or trial design versus a pure plan without any concrete data even to analyze. Of
That's
course,
great.
because of the resource, for this MIDD pilot program, we don't do the extensive reanalysis anymore like we did with the end of 2A pilot program.
Yeah,
But still, it's much more, I would say, useful to have concrete data. You already build a model, you already analyze data, and propose some concrete recommendation for the next phase. I think
great.
those kind of cases will have more chance to be accepted.
Thank you very much. And we really appreciate your presentation. We are at time now for the session. So we're going to close this session and move on to the next.